Hey everyone, yeah, welcome to our Sasonke Biotic seminar with Ersilia this evening. I'm just going to wait till everyone is admitted into the meet, and then we'll get started with a little bit of Sasonke Biotic housekeeping and just some community announcements. And yeah, then we'll jump into our talk this evening. But yeah, so let me just start by just saying a really big thank you to Gemma and Nikhil for presenting to us this evening. They're actually, Gemma's in Cameroon at the moment, so really gone out of her way to share with us. So I'm really grateful there. But yeah, just in general, some updates from what's been happening with Sasonke Biotic lately. And just to let you know, if you're here and you're interested to get involved, how one can get involved, I'm going to quickly run through that. So what's been going on at Sasonke Biotic? So two really interesting pieces of news is, one, we're hosting the Africa Machine Learning and Health workshop at the Deep Learning Indaba at the end of August this year. So if you're going to be at the Deep Learning Indaba and you're interested in healthcare, please come join the workshop. Also, if you've been accepted and you're a Sasonke Biotic community member, we're going to be working very hard in the next month and a half to get funding for everyone. We've also had a soft confirmation from the Indaba X South Africa that they'll be able to, they may be able to help us route some funds there, because Sasonke Biotic doesn't yet have a legal entity associated with it, but luckily we have some friends in this space. So both the Deep Learning Indaba itself, which is a charity that is based in the UK, and the Indaba X, which is a non-for-profit which is based in South Africa, or the Indaba X South Africa, have both offered to help us route funds to get community members to the Deep Learning Indaba. So if that is you, we will be reaching out to you soon. I think we've got about a list of 10 people that we're going to try to fund to get there. But it looks like we will be able to refund flight tickets. Not that we have the funds yet, but we have the the routing to at least make that possible. So that's really nice. But yes, so if you're here for the very first time, welcome. And if you're interested in how to get involved in Sasonke Biotic activities, there are a variety of different ways to participate. Essentially, what we try and do is make participating in machine learning and health in Africa as simple as possible. And even by coming to one of these seminars and asking questions and getting involved and getting to know the community, that is a form of participation. And if that's all you want to do, that's perfect. If you want to get a little bit more involved, sorry, you can head over to our website, which is sasonkebiotic.africa, and you'll see we have a page here with initiatives and projects. So just a quick intro to what this means. So our initiatives are essentially just general themes along which we try and organize our community. And we've identified that these are things that we'd like to try and achieve. These are really open-ended and ongoing. And then projects are our basic unit of work. And we try and then align those projects with initiatives. We've got some ongoing projects, but if you want to put your own project and you have an idea that you'd like to get help with from the community, you're more than welcome to. We have a channel on our Discord dedicated just to that, with some advice on how to go about pitching those projects. Yeah, so the two places that we're really looking for participation at the moment is we have an open project on a survey paper for machine learning and health in Africa. So that we're trying to build that up from a traditional survey paper, but then we're going to use, we're trying to use our space as independent researchers to tell a uniquely African story through that as well. So if you're interested in getting on board, we're trying to learn from what Masekane did. And if you don't know who Masekane are, I would say Google Masekane NLP. We have branched off from their community model. So We have a couple of their members here this evening who are helping us on this paper, and I think they've written a couple large collaborative papers with something on the order of like 20 to 40 authors, and that's sort of what we're going for here. So if that interests you, we're looking for junior researchers, senior researchers, people from machine learning backgrounds, people from healthcare backgrounds. We're really trying to take advantage of diversity in the space to write this paper. And another project that's we're looking for individuals to get involved is using privacy-preserving AI to make African health data sets more accessible. So entry points for all of these projects are essentially come to our weekly meet and just get involved. There's a lot of chaos, but it's lots of fun, and that's the best way I can recommend doing that. We already had 2 papers published using this method, so at least we know that the method works. And a big congrats to our community members who were able to pull this off. Go check those papers out. I highly recommend it. And also, if you'd like to jump in on any of these projects, the Discord channels on those are very active, and I'm sure our community members would be happy to assist onboarding there. But yeah, that's enough from myself. I'm going to hand over to our guest this evening. I'm going to ask them to do a screen share. And just so everyone knows, this evening we are recording, and this recording will be available on our websites, some days after this, or it'll be immediately available on Cassini. So yeah, without further ado, please take it away.
Thank you, Chris, and hello, everyone. It's great to be here presenting, as Chris has introduced. Today I'm Gemma, and I'm here together with my co-founder, Mikel. We founded the Ersilia Open Source Initiative, a nonprofit initiative for developing machine learning tools for drug discovery and global health. And we're very happy to be presenting today. So, Mikel, if you can move on. So today we'll briefly introduce our nonprofit initiative, which has a lot of similarities and interests with CIS Biotic, and then we'll go on towards defining our main project, which is this hub of machine learning models for drug discovery. And then we'll also take the opportunity to introduce a few of the concepts we are using for building this hub and our machine learning tools. Hopefully, this will be useful to some of the folks here in the audience. And finally, we'll give an overview of a project we are currently involved in, taking advantage that we are actually here in person in Cameroon. And yeah, before starting, I just want to mention, the same as Chris did, some announcements. We are also giving a workshop on data science for drug discovery in South Africa in person. There is research grants available to participate in this seminar. And we just got awarded the confirmation today that this is happening at the end of September. So we'll be sure to share the invitation with Chris to distribute to the community. So if any of the things we are sharing today sounds interesting to you, be sure to come to this extended workshop in South Africa in Cape Town. Okay, without further ado, the Ersilia Open Source Initiative is a nonprofit organization based off the UK. And the mission is to strengthen the research capacity against infectious and neglected diseases in low-resource countries, with the aim to provide more egalitarian access to healthcare worldwide. Why are we working on this space? I'm sure you're familiar with this map. This is the world representing each country according to its land size. And here you can see the classification of countries according to their income. But when we plot instead of the land area, the burden of communicable diseases, meaning infections, we see how these disproportionately affect low and lower middle income countries, particularly those countries in Africa. Whereas when we plot the number of scientific publications as a measure of the scientific output of these countries, we see how the map is completely reversed. So those countries suffering more from communicable diseases don't have enough research capacity to give a response to the needs of their population. To put some numbers into this, less than 5% of the research comes from low-income countries, but 6 out of the top 10 causes of death on these countries are still infectious diseases compared to only 1 or 2 in high-income countries. And only 10% of the drugs that are being developed worldwide are actually targeting infections, which means there is a large part of the population in the world whose healthcare needs are neglected by the pharmaceutical industry. So this is where Ersilia comes in. We try to support the development of capacity in country to do research against those diseases. So our work is based on 3 pillars. First, we work completely in the open source domain. So everything we do, we try to publish real time to share the code that we are developing. All the code we develop is eventually shared under permissive licenses, meaning everyone can access it. It's not patented. And we do so hoping that it's reproducible and easy to use by anyone. We also focus on in-country research, meaning that the scientific leadership of the projects we engage with is usually led by local institutions, our partners in different countries in Africa, and we favor implementation in situ of the tools we develop to make sure that our collaborations are sustainable, so that scientists in our partner institutions can continue the projects even after the collaboration with Ersilia is finished. And finally, we are also focused on developing tools that can be used in low resource settings, meaning low computational requirements and internet or connection bandwidth. So we started by using AI and machine learning because that's obviously it's a very hype area, but that's not the reason behind it. So basically, when you apply machine learning tools to drug discovery and to science in general, the goal is to reduce the number of experiments you need to do. So testing molecules in the computer before doing the experiments, and to reduce the time to actually find new drugs and the cost of finding them. So it's a very cost-effective method. It's faster to implement than experimental research. You can work remotely, and it's fairly domain agnostic, meaning that we can apply it to a broad range of problems in healthcare research. We started focusing on drug discovery, though you'll see in the next slides that we are also targeting other areas such as epidemiology. But basically we started on drug discovery because that's our background as scientists before starting Ersilia. And we are focused on supporting research into infectious and neglected diseases with the goal of finding new and cheaper medications than the current existing ones. Next slide. Okay, so let me introduce Ersilia, which is a hub of machine learning models. So for those of you that may not be so familiar with quantitative structural activity relations, so basically these machine learning models to predict the activity of compounds against, for example, a pathogen such as E. coli. So what usually happens experimentally is that you have a set of compounds. These compounds are incubated with the bacteria or the malaria parasite or any other organism of interest. And basically you observe whether the parasite is able to grow in the presence of the drug or not. So you get hits, whether these are active inhibitors of the bacterial growth or inactive inhibitors. This is our starting point. This is the raw data that we get from our experimental collaborators or from public databases. And then what we do is we take this data, we take these chemical structures, and we first, and very importantly, featurize them. A lot of the work that we do is based on improving this featurization, which is converting these molecules into vectors so that the computer can actually understand and integrate all the information about these molecules. So once we have this vectorial format representation of the molecules, we can then train a neural network that will learn whether a molecule is an active or inactive based on its chemical and on its chemical properties. So that when we come with a new molecule, such as the one depicted here, we also convert this molecule to a vector, and then we ask the model to predict whether this molecule will be an active or inactive. In this very simplified example, you can see how the vector is actually similar to that one of the first molecule of the training set. So if two molecules are very similar structurally and chemically, it means that they are probably both of them actives against the pathogen. So that's what the model will predict. So this is all great. What is actually happening inside all this is that you need to start by raw data. You need to process it, train your model, test and validate that it's working, and hopefully validate it externally if you have experimental collaborators. But what is lacking in the health sciences domain is this last layer, the dark purple layer, which is what we call deployment, meaning that these models are actually easy to use and ready to use by any scientist, not necessarily computer scientists with coding and programming expertise. And this is really lacking in academic research, particularly today. So many scientists cannot really use these tools that are being developed because they cannot access them. So even though they may be open source, they are not really implemented and easy to use. So what we are trying to do is to incorporate this deployment layer to a set of machine learning models so that any scientist can come with a question, for example, a drug, a new compound, select the model of interest, and then input it to the model, and without having to know everything that's going on inside, getting an answer, for example, what target is this new compound may be useful for. So in this hub of machine learning models that we are building and that is already ready to use, we have models that are directly from literature, so models that are open source that have been published in academic papers, but that lack this easy deployment layer. So we take this code and we bundle it within our hub. So for example, here we have a large effort on antibiotic drug discovery. So this model is able to predict antibiotic activity against E. coli precisely. And now a researcher can simply input a drug and get whether it's active or inactive. We also have models that we have trained at Ersilia using public data sets. In this example, you can see a large screening of antimalarial drugs. So we take this antimalarial data set, experimental data set, and we use it to train a model that will now predict whether a new molecule is an active or inactive antimalarial. And finally, perhaps most interesting, we do develop models in collaboration with our partners, with different organizations across the continent. And for example, this collaboration we are currently starting at the University of WEA on natural productivity, so that our partners can come with their experimental data and get predictions on whether new compounds will be active or inactive in their respective assays. So this is an overview of the hub. This is how it looks like as of today. So there is a number of models that you can select and run. It is not yet available online, so you do need to download the models in your own server computers, but they run in any normal laptop, so you don't need any special requirement to run these models. So with that, I think I'll handle the word to Miquel, who will give a bit more of in-depth overview of how we build these models. Thank you.
Thank you, Gemma. So, hi everyone. So I'm the person behind developing these machine learning models, mainly focused on drug discovery, as Gemma has explained. And I'm not sure how familiar is this audience with the field of drug discovery and how we apply machine learning to the problem of discovering new potential drugs for the diseases that are of our interest. So I will start with the basis. The most important thing when it comes to drug discovery is, as we know, knowledge representation. Yeah, and typically, traditionally in drug discovery, knowledge representation has been done in a structure-based way. So very explicit, very visually informative, right? So for instance, here you can see a protein, and you can see here in red, and you can see that would be the target, and you can see here a small molecule here in blue. And by representing the interaction between the protein and the small molecule, we can make the prediction that this molecule will be able to inhibit this particular protein. Okay, so this is how drug discovery has been done with the physics-based approach, if you want, for many years. When it comes to machine learning and machine learning applied to drug discovery, that is a data-driven approach, actually knowledge representation becomes, as we know, much more abstract. Yeah. In this case, we do not have such a visual way of understanding what's going on in our models, and we have to come up with alternative strategies to kind of make sense of what's going on inside these models. So let me just explain very briefly how we do it in drug discovery. Typically, the easiest way of featurizing a small molecule so that it can be used as an input for a model would be to define a vector, a binary vector to simplify it, that enumerates the structural features that we can find in this small molecule. And by structural features, I mean, for instance, a bond between a carbon atom here and a fluoride, or a bond between a carbon atom here and a chloride, or a hydroxyl here, or a tertiary nitrogen, right? So by looking at this molecule and enumerating all the features that we can find, we can map those features on a vector where each position corresponds to a feature, and we can put a 1 if this feature is present in the molecule, and a 0 if the feature is not present, okay? So this vector here, it's a mathematical representation that we can use in a tabular model that represents this drawing here. Yeah, so we go from something that a chemist can draw, a chemist can understand, to something that the computer can understand as an input for machine learning. But of course, as we know, we are today in the era of big data, or at least large data sets. Certainly, we are in this era when it comes to biomedicine. And actually, if we query this molecule in the public domain, we will learn much more than just its chemical structure. Actually, just by Googling this molecule, I can find that this molecule belongs to a certain family called phenanthrene. That's a chemical family. I know that this molecule, thanks to this search on the public domain, I know that this molecule is a potential antimalarial compound. I even know the mechanism of action of this molecule. In this case, it's the blood schizontide. I know that this molecule has some side effects like cardiotoxicity or skin itching, etc., etc., etc. Right? So, of course, one of the questions that we asked a while ago is whether we can embed all of this information in a format that is also useful as an input for any machine learning model that we want to build downstream. Okay, and actually doing this procedure, coming from the data that we can collect from the public domain into a vectorial format that represents this data, is what we call the chemical checker, and this is what I will present in the next couple of slides. Okay, so this arrow here, the procedure of converting all of this data that is available in the public domain in this vectorial vector has been the work of my postdoc days until 2021. So the chemical checker is basically a large repository of signatures that belong to different categories of information that we can find in the public domain. So here you can see a very small snapshot of the chemical checker, where we are collecting chemical information of the molecules, that is the chemical structure as I was defining before, but also target information, that is what proteins interact with our molecules. Also network information. If you are a biologist, you will know that proteins belong to networks and pathways. So what pathways might be affected when these molecules interact with our targets. Cell-based information, that is cell-based assays that we can collect from the public domain that express, for instance, which cells change their morphology when they are exposed to certain molecules, which cells die, which cells survive, etc. And also clinical information when available, that is what diseases can be treated with a certain molecule and what diseases can be caused by a certain molecule. That is what toxicology events can we observe and what side effects can we observe, okay? So traditionally, as I described before, we would have only chemical information for the molecules. Now, thanks to this chemical checker approach, we are able to collect much more knowledge about these molecules, okay? Here you can see I'm stratifying molecules by different disease categories, and you can readily see that depending on the disease category, we observe some patterns. That's a good indication, as you know, that this knowledge representation strategy might be informative downstream for machine learning modeling, okay? Because the fact that we observe patterns, it means that perhaps we will be able to build predictive models downstream. All right, so how do we assemble all of this information in a unified resource, and how can we do this systematically and periodically? Because data is uploaded all the time in the public domain. The way we do it is by using we call it a biomedical knowledge graph. And if you are familiar with these graph databases, the idea is quite simple. We have a metagraph that kind of defines the structure that we want to capture. In this case, this is, let's say, the structure of biology. We have genes in the middle. Genes are associated with diseases. Genes are expressed in cells. Cells constitute tissues. Diseases are localized in tissues. We have compounds or small molecules that are able to interact with genes. We have compounds that can treat diseases. We have compounds that cause disease, etc., etc., etc. Right? So once we have defined this metagraph, basically all we have to do is to browse the public domain and try to collect evidence for edges within this network. Okay, and here I'm just showing a very tiny snapshot of the database that we currently have. Here I think I'm showing only 50,000 edges. At the moment, we have a database that contains around 70 million edges. Okay, so it's a huge resource of data that we systematically collect from the public domain, belonging to genes, compounds, diseases, cells, etc. As you can imagine, having this in a systematic way and automatizing all of this has been a painful procedure, but now this is something that we have implemented. And at the end of the day, we can come up with a high-level view of this database that can be structured in this checkerboard. Therefore, the name chemical checker, where for each of the molecules, we can collect chemical information, also target information, network information, cell information, and clinical information, as I was describing before. And even within these levels, we have, let's say, different sub-levels of information or different flavors or scopes of the data. For instance, for the chemistry, we can capture the 2D structure of the molecules, but also the 3D structure or the physicochemical properties. When it comes to targets, we can capture the intended targets of the molecules, but also the off-targets or unintended targets. When it comes to cells, we can capture, for instance, the gene expression profile. I don't know if you are familiar with this concept, that is the genes that go up or are upregulated or downregulated when cells are exposed to certain molecules. We can also capture morphological changes, for instance. And when we go to the clinics, as you can imagine, we can capture indications, that is intended effects of molecules, and also unintended effects or side effects, which might involve toxicology. Okay. So as of now, the chemical checker contains about 1 million compounds, and you can check the resource in these two URLs, and of course all the data is open access and ready to be downloaded. We published the chemical checker in 2020 in Nature Biotech, and ever since we've been using this resource in multiple exercises. One of them was a dream challenge for the prediction of kinase activity of a panel of compounds, and we were one of the winning teams. So what can we do with these, let's say, signatures, biological signatures that we are collecting for compounds? The easiest thing we can do is similarity searches, right? So because we have vectors, we can just measure distance between vectors, and by measuring distance, we can define similarities, right? So molecules that are at short distances, they are similar from the perspective that we are measuring. In this case, I'm showing here 3 plots. Those are projections of these vectors where 2 molecules are nearby if they are similar according to this particular space, right? So here I'm plotting the transcriptional response space. Those are molecules that upregulate and downregulate similar genes in cells. Here I'm plotting the cell sensitivity profiles. This means molecules that kill the same types of cells, okay? And finally, I'm plotting here the chemical structure space. This means molecules that are similar or different in their chemical structure. And you can see here, I'm highlighting 4 molecules that seem to have the same effects in cells, as you can see, right? Because they are nearby in these spaces, but they do have very different chemical structures, yeah? And actually, even if you're not a chemist, I'm sure you can tell that those structures are not related. They are very dissimilar. However, they seem to have the same effect on human cells, right? And indeed, when we check these molecules in more detail, they do have the same mechanism of action. All of them, they interact directly with DNA, okay? All right, we can do the exact opposite approach. In this case, I'm highlighting here two families of antibiotics. And in this case, these families of antibiotics, they do have very similar structures. They indeed have the same mechanism of action. Therefore, they are clustered in the space. However, when we look at them in the human cells, they elicit very, very, very different responses. This might explain why these antibiotics, depending on the substituents they have, depending on the versions of them that we have, they might elicit more or less side effects, right? Even if they look similar chemically, even if they interact with the same target in the bacteria, the cells respond very differently to them depending on the variations that we introduce in these antibiotics, okay? All right, so I hope all of this sounds good in terms of helping research from a computational perspective. However, the chemical checker has a huge limitation, which is this concept of chemical space. So if you are not familiar with the concept of chemical space, that's the idea that there are many molecules that we can potentially synthesize, many molecules. It's actually 10 to the 60 or 10 to the 63. That's the estimation of number of molecules that we could potentially synthesize in the lab. So that's huge and unattainable. However, the molecules that we have so far managed to synthesize are only a few thousands, and test in the lab are only a few thousand. So there's a huge gap between all the molecules that we might want to explore potentially and the molecules for which we have sufficient information to build these signatures that I have been describing. Yeah. So as modelers in chemoinformatics and in computational pharmacology, we are faced with this problem that I like to represent graphically with this kind of map. This map here shows the vision of the world that the Romans had, right? And in their vision of the world, I would say that they had a pretty accurate view of the Mare Nostrum or the Mediterranean Sea, right? But when it comes to other regions of the world that they actually didn't explore enough, their accuracy was very low, okay? So we are having exactly the same problem when it comes to exploring the chemical space. There is a subsection of the chemical space, which is the chemical space that we have been characterizing in the lab, for which we have good accuracy in our models. But when we go outside this well-known chemical space, the accuracy of our models drops dramatically, okay? Actually, a more honest view of the chemical checker would be something like this, where we always know the chemical data, because chemistry information is easy to collect for all of these molecules. But when it comes to biochemical and experimental data in the biology labs or even in the clinics, the amount of information that we have drops a lot, right? Actually, when we think about the clinics, only a few thousand compounds have been tested in clinical trials, right? So there's a huge gap between the number of chemical information that we have and the amount of clinical data and also cell-based data that we have in the public domain. So one of the things that we have been working on quite intensively in the last couple of years has been in trying to impute as much information as we can in these missing gaps in the chemical checker. And the strategy that we chose, and I'm sure you're familiar with this kind of network architectures, were Siamese neural networks. The idea behind Siamese neural networks, as you might know, is basically learning new metrics, new ways of comparing entities, okay? And the concept behind the way we applied Siamese neural networks is, okay, well, we don't have all the information for all the molecules, but let's just measure similarities between molecules whenever we have information. And this perhaps will be helpful for us to infer new similarities in new spaces that we haven't observed yet in the lab, okay? So building this Siamese neural network was something that took us like over a year, and the training time in a supercomputing center in Barcelona was almost 2 weeks, yeah? So huge computational effort. At the end of the day, we could come up with a Siamese neural network that was satisfactory. The idea, as I said before, is we depart from incomplete data for small molecules. Typically, we have chemistry, and then we also have some anecdotal experimental results from other spaces. And by applying this Siamese neural network, we want to impute the signatures wherever they are missing. Okay, we try not to be too naive. We know that this is a huge prediction effort, and we know that with each prediction it comes uncertainty and error. So the first thing that we did, and we did that quite intensively, was to measure and try to quantify the accuracy of our predictions. And as you can see here, the accuracy is very variable depending on the type of data that we are trying to predict. For instance, if you focus here in this purple region here, this belongs to the binding data, and we are actually quite good at predicting the targets of small molecules, and therefore the signatures that we can derive from there are quite accurate. However, when we are trying to predict, for instance, transcriptional data, that is the genes that go up and go down, that's a much more complex exercise, and the accuracy of the predictions is much lower. Okay. Not only this, as you know, accuracy is not the only metric that we need to monitor in machine learning. We also try to assess the confidence of our predictions, and the way we do that is through exploring let's say the similarity space and this a 2D view of these signatures, right? The projection in the 2D space, where we can identify regions that are more confident. So even in the binding predictions that were usually quite good, we identify regions here in red that are not so confident. Those are molecules that are outside the well-known chemical space, whereas we see blue regions that are for which we can predict the targets with more confidence. Here you see red regions, for instance, for the transcriptional space, where most of the predictions are actually not with high confidence, as you can see here. Okay, so we did all these internal assessments. We did all the cross validations that we considered to be necessary. What's important at the end is to ask the question whether this is useful or not. So we are trying to do a lot of prospective validations. And the first thing we can do is just take these signatures that we have been deriving and we have been inferring with this Siamese neural network, project them in the 2D space and see whether these projections make any sense at all at the eyes of a biologist. So for instance, in this first row here, you can see drug molecules. Each dot here is a drug molecule. 2 drug molecules are nearby if they have similar signatures according to the chemical checker. And then I'm just stratifying here by different types of diseases, right? And you can see very clearly that infectious diseases, for instance, here, they belong to a certain region of the chemical checker space, let's say. Psychiatry molecules, they belong to a very specific region. Oncology drugs as well, gastroenterology, etc., right? This does not only apply to drug molecules. If we focus, for instance, on species metabolites, you can also see that depending on the species, we see metabolites in different regions of the chemical checker space. Even within metabolites of human, if we focus on different tissues, we can see that metabolites belong to different specific regions of the chemical space. For instance, here you can see saliva or the cerebrospinal fluid, they belong to specific regions. Or even if we just check fruit ingredients, for instance, of meat or coffee, etc., we see that depending on the ingredients, we identify specific regions, right? So the take-home message here would be, okay, we have these signatures that seem to make sense from a biological perspective because they are able to organize the chemical space in a way that is biologically relevant, as we can see here in this stratification. Okay. Of course, the other thing that we can do with these signatures is consider them as an input for a transfer learning exercise. I'm pretty sure everyone has heard about this transfer learning approach. The idea is that you collect data from a large data set and you use this pre-trained model as a starting point for making fine-tuned predictions for a data set that is specific to your case of interest, right? So we basically wanted to know whether transfer learning with these chemical checker signatures could be useful in the context of virtual screening in a drug discovery unit. And for this, we traveled to the University of Cape Town, where there is this center called H3D. H3D have been collecting a lot of data on anti-malarial and anti-tubercular activity of compounds over the years. At the moment, they have about 13,000 molecules screened against the Plasmodium falciparum, which, as you know, is the parasite that causes malaria, also M. tuberculosis, and also the escape pathogens, which are drug-resistant parasites. Okay, so they do have a lot of experimental data on whole cell assays against these against these parasites. They also have some cytotoxicity assessment of molecules, and they also have some absorption and metabolism assays for molecules that they consider to be interest, right? So they call it a screening cascade, where molecules progress if they become successful in each of these assays, right? So bringing a molecule all the way down this cascade is expensive. It's actually very expensive. And our goal as modelers is to provide a complementary virtual cascade, we call it, where molecules can be previously to doing the actual experiment, they can be assessed with the computer so that we can hopefully increase the hit rate across this cascade, okay? We did that systematically over the course of 3 months. We've been building models based on their data and using the chemical checker as a starting point. So here you can see a little bit of a benchmark where the chemical checker approach is depicted in blue, and the best possible approach that we could find only by using chemical information is depicted in red. And as you can see, especially in the orange region here, which belongs to the more, we call it physiology-based assays, that's the assays that are not like so molecular, but they involve like the whole body response. In this case, the chemical checker, this transfer learning approach, brings a substantial improvement in performance. So let's focus, for instance, in this subplot here, which is measuring the performance of the models that we built in different toxicity assays, right? So here, what we're trying to do is to predict the toxicity of small molecules. In blue, as I said, you see our models. In red, you see the models that we can build only by using chemical information. And as you can see in some of the sub-assays of this toxicity screening, the gain in performance is quite significant, is substantial. Not only this, you can also see that even if we keep removing data from our training set, the robustness of the models that we obtain stays higher in the case of models built with the transfer learning approach, which in a way suggests that possibly we could build models of good enough performance with less data, right? And thereby decreasing the cost that is needed to build proficient machine learning models in this context. All right, so what other things can we do with this type of signatures, and how have we been using them? Well, the other aspect of drug discovery that is really trendy and hopefully is going to be very useful in the next years is this idea of generative models. So in the case of a generative model, we are actually trying to do the opposite approach. We start with an activity that we want to achieve. For instance, in the case that Gemma was describing, that would be the activity against E. coli, and we want to invent molecules that will be able to fulfill this activity, right? So building generative models, as you might imagine, is much more tricky than just building a supervised predictive model, like a classifier, for instance, a binary classifier, but it can be done today in chemoinformatics thanks to different graph or language-based models that that people have been developing and we have been adopting. Let me just show you an example. Here, we applied a reinforcement learning approach based on a graph representation of molecules. And you can see here molecules that are being generated by this network. You can see here a common substructure that is kind of selected by this generator. And then you can see here in the branches different variations of these molecules, right? And the more blue the background, the higher the activity that we predict to be for this molecule. So in this way, we are able to generate a lot of candidates that hopefully synthetic chemists can consider and synthesize and then eventually test in the lab. So the result of these generative models would be in our hands are maps like this one, where you see, I think in this case, it's about 100,000 molecules that we have generated, and we color them by predicted activity, the interesting region, are those regions that are red, where we identify molecules that we predict that they will have high activity against, in this case, the malaria parasite, which is what we're trying to predict, okay? And one of those molecules would be something like this. Here you can recognize the pattern that I was highlighting before and the two substituents, okay? So by browsing this huge space of invented molecules by degenerative models, the chemists that we collaborate with, for instance, Dr. Matthew Todd at the University College London, they can actually synthesize those molecules and test them in the lab and see whether our predictions make sense at all. And this is something that is happening as we speak. They have selected from this big map that I've just presented, they have selected, I think it's 25 compounds. They are synthesizing them and they will test them in the lab in the next couple of weeks. And we are, of course, hoping to get good results, at least for some of these candidates. Remember, these molecules have been invented by these generative models. Right, so that's enough, I think, for like the kind of methods that we are applying, AI/ML methods that we are applying to drug discovery. I just want to briefly mention or enumerate a few of the projects that we are doing, especially in Africa today, right? So the first one is one that I have already mentioned, is the implementation of a virtuous drug screening cascade at H3D in South Africa. A very similar project is the one that we are currently implementing here in the University of Buea in Cameroon. In this case, the type of molecules that we are interested are natural products that they have been identified in medicinal plants across Central Africa. And the kind of activities that we want to predict is not anti-malaria or anti-tubercular activities, it's more antivirals. And the challenge here is basically to, again, like very similar to what we had at H3D, it's trying to deploy the machine learning models that we have developing so that scientists here, they can use it without having data science expertise. Another project that we are implementing, it's in Zambia, in the city of Lusaka, and in this case, it's not so much related to drug discovery. What we are trying to do is record linkage of medical data. You might be familiar with this concept. Record linkage is basically trying to link two databases that have been generated from two different entities, yeah, and you're trying to find common records between them, right? And in this case, this means trying to link HIV patients that are in one database to cervical cancer screening data that are in other databases. And this might sound easy, but it's not so easy when people do not have the same identifiers consistently in the two databases, okay? And this is something that is aided by machine learning, so this linkage is done probabilistically, actually. Another project we are doing is in Mozambique, and in this case, we are applying Bayesian methods to try to infer what is the chain of events that has led to children's death. And the input data in this case is minimally invasive autopsies because of lack of resources, and also because of ethnical constraints. Sometimes the full autopsy are not allowed. So our collaborators in Mozambique, they are collecting, they call it minimally invasive autopsies, and the goal of our methods is to actually predict what was the actual cause of death of these children based on this minimally invasive autopsy data. All right, so, and also the open source antimalarials data, this I have already presented. This is generating new molecules based on generative models, and then these molecules are eventually tested by our collaborators. And finally, one incipient project that we will start implementing, again at H3D in South Africa, is the idea of optimizing the doses that are being given, especially with in tuberculosis patients, adjusted to the genetic background of African ethnicities. Okay, as you may know, many of the drugs have been optimized with Caucasian population, even anti-tubercular drugs, and some adjustments would be necessary to adjust the therapeutic window and also reduce the number of side effects. All right, so I think that's all. I would say that the take-home message are the following. We are Ersilia. We are not-for-profit organization, and our mission, as Gemma has explained, is to democratize access to AI/ML tools in biomedical research. We're trying to build, as you are, an international network of collaborators in the field of AI/ML and also research in biomedicine. We combine remote working with on-site project development. That's why we are here now in Cameroon, for instance. All of our assets are obviously open source because we believe in this model, and we work at the intersection between academia, startups, and pharmaceutical companies. And of course, we are always welcoming new contributors and collaborators. And that's all, I think. Thank you very much. And we are happy to take questions.
Cool. Thank you so much. I mean, I'm definitely going to watch the recording of this video again because you guys went into some like really beautiful depth on what you're doing there. I think what you're doing is absolutely fantastic. I think everyone here will agree that an open source model definitely helps innovation and accessibility. So I'm going to invite our audience to ask questions. You can pop a question in the chat. You can, if you'd like to ask your question in person, just ask you to use your reactions at the bottom of your Zoom to just raise a hand, and then I will invite you to ask a question, and you're welcome to unmute your microphone, or if you'd like, you can turn on your video. I will prioritize some of the Masikane individuals we have here because I know you guys have been meeting at 7. But yeah, let's kick it off. I see Chris Emuzu already has a question. Chris, would you like to ask that question? You're welcome to go ahead.
I already answered, I think. So yeah, Chris, so may I read the question? Yeah, so basically Chris is asking whether we just use one Siamese neural network or we actually used multiple neural networks depending on the space that we were trying to predict. And this is indeed the case. We actually trained 25 Siamese neural networks. If you remember, we had 25 spaces in this checkerboard, and there is one Siamese neural network dedicated to each of the spaces. So thanks for this very fine question. Thank you.
Sorry, I have a follow-up question to that. What happens if, like, because they're kind of connected, right? So you have the chemical components and you have the clinical with different components. What happens if you have a model that kind of, because if you have different networks, they're only taking in isolated information for the task. But if you have a model that is kind of using many of them, the features, was there some studies on that?
Yeah, so it's we tried to do that, but because the amount of data that we have in each of the spaces there's so much imbalance that for us it was quite challenging to actually like to give the same weight to each of the data sets. Okay, so the model was actually just learning the things that were most represented, yeah. Okay. And therefore, at the end we decided to train independent neural networks.
Okay, yeah. Thank you very much.
While I wait for more questions from the audience, I'm going to ask you one or two of my own. So I see, I mean, clearly your focus is on drug discovery. Are there plans to move to anything beyond just drug discovery? Other spaces like diagnostics or other treatment vectors?
Mm-hmm. Yes, thank you for the question, Chris. So definitely we started by drug discovery because that's where the models are easier to automatize. So we have a set of automatic ML tools that we are using for all these collaborative projects. We are certainly looking into epidemiological type of studies, such as the ones that Mikael was sharing in Zambia and Mozambique. And we are also trying to start some projects more on the diagnostic side. But in this case, the tools are not so automatic, so it's very much project-based. Mikael, do you want to?
Yeah, and I think I should also mention that within the field of drug discovery, we have an interesting project. I hope it's interesting on privacy preserving AI. And because you've mentioned that before, I think that that would be something to follow up on. Okay, the idea here is not to share patient data, but how to share small molecule data within pharmaceutical companies. Yeah, because of course pharmaceutical companies want to keep the information of the compounds that they have, but at the same time it would be very interesting to leverage all the assay data that they have historically been collecting, right? So a privacy preserving approach to that would be quite interesting in terms of unlocking all of these data sets that are currently like just hosted by the pharmaceutical companies.
Cool, thank you. My second question was actually going to be around data privacy. And just before I get into that, just to let the audience know, because I mean, our audience is generally either from a machine learning background or healthcare. So please feel free to ask any and all questions. All questions are good questions. I know there's quite a lot of technical talk going on at the moment, but if you have any other more broad questions, please feel free to ask. So yeah, data privacy is, it's a huge topic in healthcare, and as it should be, because we need to protect data privacy, and especially in healthcare. So, I mean, you mentioned that you work with partner organizations. I'm curious to understand when you, so you mentioned there's sort of two ways you get your data. One is your systematic sort of scraping of what's out there. And I'll actually add a second question to that on there. But for the partner organizations that you do work with, how do you guys navigate that data privacy is my first question. And then my second one is another big topic in healthcare ML or AI is around safety. So when you are doing this systematic scraping, how do you go about validating or ensuring that what you're producing is sort of safe knowledge? Sort of that.
Yeah, thank you, Chris. So for the first question, the answer is easy. At this moment, we don't have the capacity or the knowledge to, for example, anonymize patient data and so on. So we rely on our partners. So this is the IP of the data is dependent on our partners, and they retain it. So in some occasions, we are not able then to share the data sets that we have used to train the models. That's something you have to live with, right? So basically at this stage, we ask our partners to take the burden on protecting the IP of their assets, and for ensuring the quality of the assets. That's a really good question. So for example, when we take assets developed by others at this moment, we try to only incorporate assets that have been published and peer-reviewed by others, so we can assure their quality. And we are still deciding how to deal with other types of assets that may be very useful but are not published. So how can we actually ensure that they are of quality enough? And for what we produce, we use the typical benchmarks, for example, in drug discovery against MoleculeNet or other benchmark data sets.
Yeah, thanks, Gemma. I think I would like to add here in terms of safety that we work on at the preclinical level. So we just make predictions. We hope that these predictions are useful, but there's a lot that needs to be tested before actually delivering the small molecules that we are predicting to the clinics, right? So actually our goal is to reduce the attrition rate of experiments that our collaborators do. It's not actually just to suggest a molecule that we can deliver right away to a patient. That's really not our goal at this point. So in this sense, of course, we still need to undergo all the standard safety procedures that are thankfully implemented by the regulatory agencies, right? Yeah, and about the privacy preserving, Gemma has answered with regards to patient data. I think that with regards to intellectual property of the small molecules that are basically proprietary of our collaborators, it's really case dependent. So some collaborators are really willing to share data, Some are more, they have greater restrictions. It really depends on the case. We just decide on a case-by-case basis. What I can say is that we are trying to develop this tool for privacy preserving AI that basically releases encrypted versions of the models so that the identity of the compounds cannot be reverse engineered, and we can at least share the models that have been trained on the data without actually disclosing the identity of the compounds that have been used at training time.
Thank you. So we have 2 more questions. I was gonna invite Bolu if they would like to ask, but I see Gemma, you've already answered, so thanks there. Chris?
Okay, I have 2 things to say quickly. One is that, so I'm currently very interested and invested in the Bayesian structured learning and cause effect estimation. So I would really love to probably later on look into if there are research areas that you're working on currently and the way I could help come in, contribute. I would love that. The second thing is I'm really interested from a business perspective, like what inspired you guys to go nonprofit? Because I've seen a lot of startups with drug discovery and drug repurposing, and they are mostly always for profit. And it's a very bustling business right now when you're doing healthcare, like, you know, you have a lot of investment. So what inspired you to go in the nonprofit direction? I'm also asking because of my own, I'm really inspired by that.
Thank you, Chris. That's a great question. I think the first and most honest answer is we never considered being a for-profit. We come from academia and we believe in open source and in sharing as the only way to actually cure all these neglected diseases. So a for-profit initiative here would be more difficult to actually achieve our goals. And because we are really focused on implementing those assets in low resource settings, our end users, like researchers like here in Cameroon, if we were a for-profit company, they would certainly not be able to afford our services. Whereas as a nonprofit initiative, what we can do is we apply together for grants. And once we obtain funding from philanthropy and research projects, we can then work together. So that's the main reason, basically, that we believe that the people we are trying to help and to serve benefit more if we have a nonprofit structure than a for-profit one.
It's very inspiring. Thank you so much.
Thanks for the question.
So one more question from myself. So with what you guys are trying to do with drug discovery, that I imagine then would feed into, like you mentioned, a clinical trial process, which again feeds into safety. However, I can imagine, one of the things that it seems to me that you've done that has been really impressive is the knowledge representation and being able to take a lot of information from different sources and combine them in a meaningful and effective way. So are there any other routes that you've thought about that might have immediate implications? So as an example, one of the things I've seen in the space is just looking at certain combinations of drugs. And for instance, in a South African context, we have a large burden of HIV, and generally a HIV regimen will be 3 different types of antiretrovirals. So I can imagine a situation where towards like personalized medicine, you can use someone's gene sequence or even just other information about them, like you mentioned, and then try and find the right combination of drugs, which seems like something that's, all of those drugs are already safe enough. Is there anything like that you guys have looked into?
Yes. Yes, unfortunately, we have in the context and in the past, in the context of cancer cancer research and only in the context of cancer cell lines. So nothing that can go, let's say, directly to the clinics, and also only not drug cocktails, but only pairs of small molecules. Yeah. And the reason why is because you only get training data with like systematic exploration of pairs in the context of cancer screening. So unfortunately, this is not something that you find quite easily for pathogens, and that's a pity. Hopefully we will get there. Yeah, but I would say it will have to be through unsupervised approaches, which, as you know, it's more difficult and more challenging. But yeah, I mean, I would say that for many pathogens, especially the drug-resistant pathogens, that's the way to go, right? So yes, indeed.
Awesome.
I think Michael has the hand raised, Chris.
Yep. Go ahead. Michael, you're welcome to go ahead. Okay, Michael might be experiencing some technical difficulties while Michael, please feel free to jump in at any point. Otherwise, in the meantime, we have another question in the chat from Tassala, I think. You're welcome to unmute your mic if you'd like to ask, otherwise I'll read it for you. So
yeah, we can answer that one.
Yeah,
we can answer that one. We can answer that. So definitely, we are now looking at implementing into our models those libraries that help on explaining, for example, when you predict the activity of a molecule, what are the specific atoms and connections within this molecule that actually confer the activity. So certainly this is something we are actually currently developing. So hopefully before the end of the year, it will be implemented in our tools.
Yeah, I think the two strategies that we're using is, one is, you might know, Shapley values. Shapley, do I hear my echo? All right. So thank you. So Shapley values, as you might know, is they are used to. I mean, it's like they come from game theory. Is like additive approach, and basically what you want to do is to try to discover which features in your model are the most are the ones that are more significant in actually giving you the prediction. So Shapley values, we have been implementing them with easy to understand descriptors of the small molecules that would be physical chemical properties. Then in terms of what Gemma was mentioning, identifying the atoms or the substructures in the molecules that we believe are key to the activity that we are predicting. For this, in this case, we use a concept called counterfactuals, right? Where you make a lot of modifications of the small molecule, right? And then you explore the predictions across this very local chemical space. And then whenever you find a molecule that has a very different prediction to your original molecule, but is nonetheless very similar to it, then you have identified a critical point for the activity. So counterfactuals is combined with Shapley values is currently what we are using for interpretability. And according to our collaborators, at least, this turns out to be quite useful.
Yes, thank you. Are there any more questions? Otherwise, I'm sorry for people's time because I know we are over time.
So I'll invite Michael just to see if he's there to ask the question he wanted to ask. Maybe Michael put that in the text in the chat, but also again, I think we'll have that then as the last question, because I can imagine that traveling through the Cameroon is extremely tiring. So, or traveling in general, let's call it that, can take it out of you. So I think there's that one extension of that last question in the text, and then we'll call it an evening.
Okay. So I think I can answer this one. So the question is whether we use textual data and as input features. And I hear an echo. So thanks. So basically, the idea here is that most of the times it's actually not text data. Even if I was representing that as text data, most of the times it's structured data. Okay, most of the times. When it comes to text data, we use like pre-trained models like BioBERT and things like that to come up with embeddings of short paragraphs of biomedical text. Okay, this we have been using, for instance, for bioassay descriptions. Yeah, we don't train the language models ourselves. We use them from pre-trained sources like Cybert and things like that.
Cool, thank you so much. I mean, also, if anyone wants to do a virtual round of applause for our guests this evening, please join us in that. I must say, the work that you guys are doing is so exciting. You know, if there's, I think if anyone from our community would like to get involved, I will. What do you think is the best way, or might be the best entry points there?
So just share our emails for now. I was already speaking with Chris, the other Chris in the conversation. This month might be slower to respond because of our commitments with our partners, but certainly we'll get back to everyone as soon as possible. So just write to our email. We are very happy to answer questions and explore future projects. So.
Cool. Thank you very much. Yeah, and with that, we'll call this seminar to a close, and very glad that we were able to host you this evening.
Thank you for having us, Chris.
Yeah, it's been a pleasure. Thank you, everyone.
Sure. Thanks, everyone. All the best. Cheerio.